Hello, everyone. So it's 8 p.m. now. Let's start our meeting now. Okay. Okay. I'm Wang Xin from Professor Lin's group, and on behalf of the SyncBio community, I'm so glad that we gathered together to share our latest findings and interesting results, particularly in the systematic and evolution of syngnathid fishes. So the first presentation is provided by Professor Healy Hamilton. So firstly, I will give a brief introduction of Dr. Healy. Okay, Dr. Healy is the chief scientist at NatureServe with graduate degrees from Yale and the University of California, Berkeley. Also, Dr. Hamilton leads a staff with expertise in ecology, zoology, conservation, data science, and information management. She is also a world expert on the taxonomy and the evolution of seahorses and their relatives. So today she will present a part of her research going on in her group. So now let's welcome Dr. Healy for his presentation.
Thank you very much. Can I, good morning, everyone, for me, good afternoon, good evening, wherever you are. Can I confirm that you are seeing the correct screen? Okay, we're good, thank you.
Yeah.
Thank you. So I would very much like to thank the organizers of SyngBio 4 for the kind invitation to present to you today. And I'm sharing this presentation slot with my dear friend and colleague, Graham Short, who many of you know, and who is also a world expert on the taxonomy and evolution of seahorses and their relatives. So, today we are going to talk about, some parts of this talk are actually a little repeated from what we presented in 2017 when we had a newly published molecular phylogeny that integrated morphological characters, and that phylogenetic hypothesis of syngnathid evolution remains the most comprehensive published phylogeny that we have to date for sygnathids. But we're going to try to examine it from some new ways based on research that Graham has been leading into identification of novel morphological characters. So we will share this time slot about half each. So I think, one question we have is just, why does phylogeny matter, right? Let's make sure we understand that the reasons that we study the tree of life in the first place. So here I have a slide showing the origin of species from Charles Darwin. And there was, in that whole several hundred-page book published in the 1850s, so almost 200 years ago now, there was one single illustration among the entire book. And that one illustration is here. It was essentially a representation of ideas of evolution, ideas of tree thinking, the pattern of ancestry and descent that represents how species evolved. So from the very beginning of our understanding of evolution, from Charles Darwin himself, we looked to evolutionary trees as a way to express those patterns of ancestry and descent. And then a quote, one that many of you may well know and may have heard from really one of the greatest evolutionary thinkers of all time, Theodosius Dobzhansky. He shared with us that Nothing in biology makes sense except in light of evolution. Phylogenetic trees provide that historical narrative for us to try to understand the similarities and the differences, the morphologies, the behaviors, and the pattern, even genetic patterns. Like without that understanding of the branching pattern of the tree of life, we are missing the sort of the fundamental driver of most of the phenomena we attempt to understand about the species that are extant today and the fossils that paleontologists study as well. So that understanding of the pattern of evolution is absolutely fundamental to looking at male pregnancy, to trying to understand mating behaviors, to trying to understand the bizarre and fascinating morphological characters that we see in our particularly derived and highly mysterious group of fishes. And this is a group of fishes that has fascinated us for all of the history of man. There have been representations of syngnathid fish in tile art that's thousands of years old, in literature, in sculpture, in art. This is a group of fish that we have, that human society, whether you're a scientist or not, has been absolutely fascinated with for all of history. And so understanding the evolution of this group helps us to understand how did this group of fish that is so different from other fish, that has done more to rearrange the body plan of what it means to be a fish than any other fish group that is extant. And since the beginning of the field of systematics, we have been interested in trying to understand this particular fish group. Carolus Linnaeus in the 1700s actually included several syngnathid fish in Systema Naturae, the very first effort to catalog life on Earth. So you know, this group of fish has fascinated us from the earliest times of culture and the earliest times of our attempt to make sense of the classification of species, to make sense of the diversity of life that we have been faced with, really that we have been blessed with. So the early attempts to really try to understand how seahorses and their relatives, how syngnathid fish evolved, but mostly how they're related to one another, right? So we've been driven to understand, where do seahorses come from in particular because of their unique morphology? And so this study, it's not the first study that ever looked at syngnathid taxonomy, but this graphic from this study by Earl Harold, who was the director of the Steinhardt Aquarium, and this was published in 1959. And it's the first time that anyone ever tried to put the pattern of morphology into some kind of a branching pattern, right? So he's trying to understand essentially how seahorses arose from pipefish. And this was the earliest study of its kind, and also showing brood pouch evolution and emphasizing the importance of brood pouch evolution as a character to allow us to understand the trajectory from a pipefish to a seahorse, which he saw through pygmy pipe horses as the intermediate morphology between a pipefish and a seahorse. But it does emphasize brood pouch evolution. But really, sort of the father, if you will, of syngnathid taxonomy was this man, Chuck Dawson, who was a contemporary of Harold's and who published with Harold. And the astonishing thing about Chuck Dawson is that he had one of his eyes was made of glass, And so he sort of looked at everything with through one eye, but he used that eye to study extremely carefully syngnathid fishes, but only in his lab in Mississippi. He never went into the field to examine these fish, but he was the one that came up with the morphological characters that help us diagnose species. So he looked at at cranial features, at brood pouch features, at features of dorsal fins and tail and trunk rings. He looked very carefully at these fish, and he was able to diagnose to species literally dozens and dozens of different syngnathid fishes, but he never was able to use the characters for diagnosing species to understand the pattern through which those species arose. So he was fantastic at taxonomy, sort of the father of syngnathid taxonomy, but he was never focused on providing a hypothesis of the relatedness of the fish that he was describing in any sort of deep way. And I'll show you an example of what his challenges were. So here we've mapped onto the phylogeny we presented in 2017. We mapped some brood pouch characters. So here you're looking at the color patterns of different ways the brood pouch closed, mapped onto this hypothesis of evolutionary relationships. And what you can see is the colors flop all over the place. They come and go. If this was if these characters were diagnostic, you would want to see deep nodes that were all yellow or all blue or all black, deeper in the tree. And instead we see the opposite. We see this group of characters flip-flopping, turning on and off all across the tree. So it's an example of a morphological character that is not really helping us resolve those deeper nodes of the tree. And this has been the sort of challenge of syngnathid taxonomy, syngnathid phylogeny, and why it was so late in the science of studying syngnathid evolution that we actually came up with a more robust understanding of how these animals how they evolved, how they're related to one another, and what that tree of life, what those branching patterns really look like. And it was not until molecules and sort of being able to compare sequences of DNA among different syngnathid fish that we started understanding evolutionary patterns. And the early work, such as this first early work on genetics of sygnathids by Tony Wilson back in 2001, which was the same year that I got my doctorate, and we both studied partial mitochondrial gene fragments, right? Back then, you could actually get a PhD with a few dozen samples and a few partial mitochondrial gene fragments. And this early work by Dr. Wilson, both this work in 2001 and this work in 2003 was not necessarily trying to reconstruct the full branching pattern of the syngnathid tree of life, but was very focused on brood pouch evolution, right? Dr. Wilson studying male pregnancy, trying to understand the reproductive patterns, and mapping the morphological character of how the brood pouch closes onto a tree that was derived by molecules. And so this was sort of our first understanding of the deeper branching patterns, the deeper splits in this evolutionary tree. That was followed in 2009 by another study by Peter Teskey and his colleague, Dr. Baragaray, and they were looking at sort of the timing of major branching pattern. You can see that there's very few species that are sampled here, but there are fossils mapped onto the branching pattern of those trees. Now, these are just a handful of a few of the early studies for syngnathid evolution that used molecules and that mapped morphological characters onto a tree derived from molecules. And both of these really had both of these scientists were really looking at particular. The sampling that they had of sygnathids did not allow them to understand the sort of that comprehensive sampling that was necessary to get a more full picture of sygnathed evolution. So taxon sampling in these early efforts was really the big limitation. The genera of Hippocampus and of Syngnathus were heavily sampled, but a lot of those other weird and wild syngnathid fishes, many lineages just had not been represented yet in these early studies of syngnathid evolution. And so that is where my colleague Graham and I we come on the scene when we had some early studies of syngnathid evolution based on molecules, but just not the taxon sampling to provide us with that more comprehensive understanding. So we spent essentially 10 years from about 2007 to 2017, traveling around the world wherever we could, working in, from Australia and the Philippines to the Caribbean, working with colleagues all over the world, including Dave Harasti was helping us here in Australia. And we were successful over those many years in sampling 47 of the 56 recognized genera of sygnathids, which allowed us, really for the first time, a more comprehensive understanding of their evolution. And so this is a tree that we presented in 2017. And it was sort of hot off the press at that time. And so really, we had almost 100 species. We had 48 of those 57 genera. At the time, looking at 5,000 base pairs of DNA with 4 nuclear genes and 4 mitochondrial genes. At the time it was published, that was a big deal. If any of you saw Josephine Stiller's talk a few days ago, you'll know that this is quite outdated and even quaint today, but it is still the most comprehensive published phylogeny of syngnathid fishes at the time. So we found with this much denser taxon sampling, we identified several lineages that really require significant revision, right? We found in different parts of the tree, we found species of Micrognathus, species of Cosmocampus, species of Halicampus that are coming up in completely different clades. So we showed this really deep split between the Atlantic and the Pacific for taxa that Charles Dawson mostly had put into the same genus, but even he knew back then that there may have been issues. But really, what we're going to focus the rest of the talk on, and the most interesting results came from this crown group. Because of this dense taxon sampling, we were able to identify a completely novel and unexpected crown group of relationships that included the seahorses, the Pacific pygmy pipe horses, in this crown group, but those pipe horses did not come out as sister to seahorses. So I want to pass this off to Graham to help us understand, how real are these relationships? Is this the true pattern of evolution among these fishes? If so, we should be able to sort of see a signature of that pattern in some aspect of their morphology that would allow us to confirm or refute what we have presented based on molecules. So we want morphology and molecules to converge on similar results, right? We want an integrative approach where both lines of evidence point to the tree of life of sygnathids. So to discuss how we explored some morphological characters to evaluate these surprising molecular results, I am going to pass it off to Graham.
Thanks, Healy. So this presentation, I'm going to describe some novel morphological characters in sygnathids identified via micro-CT scans or microcomputerized tomography, which is an exciting new imaging tool for taxonomists for the generation of high-resolution 3D images of skeletons and fish specimens. Traditionally, taxonomists would stain the skeletons of fish or use microscopy to look for morphological characters in order to describe new species of fish or systematically revise currently described species. However, the micro-CT scan images can be used to look at the internal morphology of sygnathids for the identification of novel morphological characters otherwise not seen by traditional methods. Next slide, Healy. As an example, this is a CT scan showing multiple views of the Western Atlantic pipefish, Micrognathus cranitus. Here you can see well-defined trunk rings, bony ridges on the snout and on the head, and the bones that actually form the skull or neurocranium. Next slide. Likewise, this is a CT scan of showing multiple views of the Western Atlantic seahorse Hippocampus erectus. These views reveal the well-defined and well-ossified trunk area, trunk rings, and the bones that form the skull or neurocranium. Next slide. So this is a CT scan of a close-up dorsal view of the neurocranium of Micrognathus cranitus. Here you can see the neurocranium is composed of 4 principal bones. The blue dot indicates the supraoccipital. I can say SOC because it's hard to pronounce. The anterior nuchal plate indicated by the purple dot, and the posterior nuchal plate indicated by the cyan dot, and the post-temporal lobe indicated by the red dot. Examination of many pipefish species I've looked at has revealed that the presence and order of these bones are typical of pipefish. And as it turns out, the size and shape of these bones are highly informative as diagnostic characters, which allow for the identification of species at the generic and species level. And most importantly, these diagnostic characters are excellent for finding patterns of neurocranial diversity within and between clades, which can be used to help resolve phylogenetic or taxonomic ambiguities in the placement of certain genera and species in the tree that we generated. Next slide. So for example, as Healy mentioned, in our multi-gene phylogeny, the pygmy pipe horses are recovered as polyphyletic, with 2 genera, Acentrionera and Idiotropicus, recovered in crown group of Indo-Pacific pipefish, and 1 genus, Amphilicturus, recovered in the Atlantic-clade pipefish. The members of these 2 phylogenetically distant lineages look very similar in appearance. Therefore, traditional external characters such as the typical angled head, brood pouch, and prehensile tail do not resolve the paraphyly of the pygmy pipe horses. Next slide. So here I'm going to focus on the Indo-Pacific genus Acentrionera, as it's mostly similar to Amphilicturus in appearance. As you can see in the top row, you see 3 members of Acentrionera, and the bottom row, you see Amphilicturus. You can see these 2 lineages share remarkable similarities in morphologies, to the extent that Dawson, 1984, suggested that all pygmy pipe horses share a common ancestor. So considering the convergence and this external morphology in these 2 lineages, are there any internal morphological characters that can help resolve the paraphyly of the pygmy pipe horses? Next slide. So here you can see the overall view of the body plan in Acentrionera and in Amphilicturus. Top left, you see a male and female Acentrionera tentaculata. And I'm going to focus on this species because the other 2 members in this genus are very similar. And in the bottom right is Amphilicturus, male and female. They share overall body shape. They share head angle below the body axis. They both genera have male brood pouches and plates, which is remarkable, and a prehensile tail. But there's some differences already you can see between these 2 genera. Acentrionera does not have a caudal tail, whereas in Amphilicturus, there's a almost smaller vestigial caudal tail. And also in Acingenura, the dorsal fin origin is on the trunk and tail, whereas in Amphilicteris, it starts on the second tail ring. So those are obvious differences. But let's have a closer look to see what distinguishes these two genera. So what I found is that the neurocranial bones are highly diagnostic. So for example, in the typical pipefish configuration, you're going to see that the supraoccipital, or SOC, the anterior nuchal plate, posterior nuchal plate, and post-temporal lobes, they represent a typical suite of neurocranial bones found in all pipefish. In Amphilicteris, you actually see all four neurocranial bones, whereas in Acingenura, the posterior nuchal plate is missing, and that's a key difference between the two genera. Also, the shape and sizes of the SOC and the post-temporal lobes are quite different between the two genera. I won't go into too much detail, but you can see that there are quite a lot of differences. Okay, next slide. Another example is the paraphyly of Helicampus that Healy just mentioned. Helicampus recovered as paraphyletic. You can see there's multiple groups. Helicampus macrorhynchus in particular is recovered in the crown group with hippocampus. So we were quite curious about Helicampus macrorhynchus in general. So examination of the neurocranium reveals some unexpected and subtle characters that are shared with the seahorses. And as Josephine Stiller mentioned, Helicampus macrorhynchus is actually the syngnathid as sister to the seahorses. Next slide, please. So here's a typical representation of a live Helicampus macrorhynchus, and below is a CT scan of a full body of one. And as you see already, the long snout and some of the characters of the neurocranium suggest is different from other pipefish. Next slide. So this is a beautiful scan of a lateral view of Helicampus macrorhynchus on top. And I've indicated each neurocranial bone, the SOC, the nuchal plates, and the post-temporal lobe. You compare to a typical pipefish below, which is Micrognathus cranitus. The bones in Helicampus macrorhynchus, the SOC and anterior nuchal plate, the blue and purple dot, you can see that the two bones are upright and are at an angle, and they're practically leaning on top of each other and forming a gap in between those two bones. Whereas in a typical pipefish below, all those neurocranial bones are fused together. There's no indication of any upwelling of the bones to form the crevice that you see in Helicampus rhynchus. Also, the SOC in Helicampus macrorhynchus is highly sculpted. It's very unusual for pipefish to have any formation or bony formation on the SOC, except for a bony ridge. But here it's quite different. Next slide. And so here's a comparison of Helicampus macrorhynchus to Hippocampus camelopardalis from Eastern Africa. You can see in the seahorse that the SOC and the anterior nuchal plate are at an angle to each other, and there's a gap between those two bones. And it's exactly what you see in Helicampus above. And this is the only pipefish I've seen in which the SOC and the anterior nuchal plate are quite different and are leaning against each other. It's the only pipefish I've seen so far that has this, and it's a great indicator that they might share a possible ancestor, but based on genetics, they do. So a lot of work is required to continue the study to support the idea that Helicampus macrorhynchus is sister to the seahorses, but this is a very good start. So in conclusion, micro CT scans are a powerful tool to examine internal morphology of sygnathids and identify novel diagnostic characters of the neurocranium in comparison to traditional methods like microscopy. Also, CT scans combined with genetics is a rather promising integrated approach to support or refute phylogenetic relationships in sygnathids. Thank you, and shishya.
Wow, Graham, that's impressive. Thank you very much. And I know we're running, excuse me, a little bit short on time. Dr. Jin, I'm not sure if you want us to move straight ahead into the next talk, or if there's a chance for one question or so.
We can have some questions. If somebody has any questions, you can unmute yourself.
Hello, Graham.
Hi.
I'm very interested in your research about microcity because we also do some microcities in our lab. So thanks for your sharing. My question is that how do you choose the key characters to distinguish different genera or different species? Because for what we see, you have show very good characters to distinguish this genera or species. So that's my question, how to choose the key characters. Thank you.
Oh, you're welcome. So for example, in the pygmy pipeporous acintronura, the superaccipable or SOC, they're quite different from each other between species. And then in turn, they're quite different from that seen in Idiotubiscus and other pygmy pipehorses. So when I scan the 3 species of acintronura, right away I can see the differences in the SOC. They're quite different from each other. And unfortunately, you can't see it, via microscopy or fish staining. You have to use CT scans in order to see the differences. They're very minute, but they're there. They're quite distinct. So I found that meristics doesn't really help in identifying species. Tail counts, trunk counts. I mean, it helps, but it's not the end all. But if you look at neurocranial characters, it really helps in distinguishing between genera and species.
Thank you.
You're welcome.
Okay. Thanks, Dr. Healy and Dr. Graham. Thanks for your wonderful speech.